Okay, so today's webinar will focus on release 47, or the beta releases of all of our sites. And there are a number of people who are present in this webinar. The ones who will be presenting today include Evelina Bashenko, who's based at the University of Liverpool, Gloria Geraldo Calderon, who's from the University of Notre Dame, based in Colombia, actually. Myself, Omar Harb, I'm based at the University of Pennsylvania, along with David Roos, who is based at the University of Pennsylvania as well. And finally, Suzanne Warrenfels, who's based at the University of Georgia. I'm gonna go ahead and switch it over to David Roos, who's gonna kick us off with some remarks, and then we will go ahead and go into the details of the webinar. So David, I'm making you presenter right now, and you should be able to go ahead and show your screen and present. All right, we can see your screen, David, go ahead. We cannot hear you, David, if you're trying to if you're speaking already.
Sorry, Omar. Yep. Thank you very much, Omar, and welcome to everyone. It's exciting to see 241 and counting attendees joining into this this weekly webinar. Congratulations to the outreach team for the nice job that they've done in putting these webinars together. I wanted to take just 2 or 3 minutes at the beginning of today's presentation to say something about the newly merged VEuPathDB Bioinformatics Resource Center, announced at the top of the web page that you are seeing, and the beta release, which is the focus of today's webinar. This is a fully merged infrastructure bringing together various different resources that many of you are familiar with. From the poll, I could already see that there are many users of FungiDB, symbolized by the the turquoise symbol at the bottom of the page. Also, many of the parasite resources collected together in the former VEuPathDB database devoted to amoeba and cryptosporidia and giardia, a variety of apicomplexa, trichomonads and kinetoplastida, as well as arthropod vectors of disease, supported by VectorBase. And I want to emphasize that despite the merger into the VEuPathDB parent organization, all of these websites will remain active, because we recognize the importance of the self-defining nature of the communities that they support. Those websites will be available moving forward with new data, and the legacy websites will also remain available, but not updated with new data for those who are familiar with the existing look and feel. As you can see from the website, the look of the VEuPathDB and all of these component databases is substantially different now, and I want to spend just a minute or two to say why that is and what is coming, including highlighting increased emphasis on the HostDB database symbolized at the bottom, and the various other resources supporting evolutionary analysis of orthologs across species and our related sister groups associated with epidemiological data and microbiome data. The release of this website is the result of a lot of work by a substantial team of people that I believe I can show you here. The individuals are shown over on the right. These calls are being run primarily by the outreach team that Omar indicates. And the point of this busy slide is just to indicate that we're grateful to support from a variety of different sources, supporting individuals located at a variety of different places, but all organized into functional groups devoted to, say, data processing, data loading, infrastructure development, user interface development, and so on. And the last thing that I wanted to comment on today is to say something about why we are doing this and what comes next. Why we're doing this is, one answer is that the merger of these resources is mandated by our funders, who are always looking to try to improve cost efficacy, as are we. But it is also a good idea in ways of addressing the scalability and sustainability to accommodate the ongoing explosion of data sets, and gives us the opportunity to integrate a variety of functionality that you'll see today, making available, for example, some of the query architecture that have been pioneered by VEuPathDB to the fungal and vector communities, taking advantage of some of the abilities to view geolocated data and to use ontologies to define various phenotypes and locations that have been pioneered by VectorBase and so on. We realize that there's been a 6-month period now with no release of new data as we've engineered this new system, but we do expect to be returning to regular bi-monthly releases with much more data, including new data types, certainly single-cell RNA-seq data, but a variety of other new data analysis and visualization tools. This provides a common genome browser for that is compatible for visualizing genomes that you're used to, but also for updating information and for use by professional curators using the Apollo platform that someone of you will recall from the webinar run by Dan Lawson a couple of weeks back. Additional tools to summarize data, making it easier and easier for you to determine which of the many experiments that are represented tell you interesting information about, for example, transcriptional profiling data. And also, as we continue to partner with our colleagues at Ensemble, better access to population genomic data sets that will particularly help in the analysis of host pathogen interactions, improved resources for looking at pathways, whether these are metabolic pathways or systems biological networks, and additional tools in the workspaces allowing users to analyze their own data and expediting data analysis and deposition. How can you help? First and foremost, contact us with your questions, your suggestions. There is a contact us link at the top of every page. Please let us know what you need. Everyone you saw in the last image is working on this project to support you and your research. If you're interested in more about the project, in the About link at the top, you can find additional information, including capturable links if you would like to cite these resources. This is something that our funding agencies are concerned about. They want to make sure that the sites are used. They'll certainly be happy to see 254 attendees on today's webinar at the moment. So we would encourage you in your publication to indicate in the text that this data was downloaded from TriTripDB, or genes were prioritized based on analysis in FungiDB or VectorBase or ToxoDB or GiardiaDB, whatever the source may be. We'd also like to encourage you to consider including one of these logos in your acknowledgement slide for presentations, much as you would acknowledge the investigators who provided you with reagents such as antibodies for your studies. So with that, let me turn it back over to Omar and the outreach team to kick off the introduction to the VEuPathDB beta sites.
Thank you, David. I think there were a couple of questions coming in. I think a couple of people may have had some audio issues. Let's hope these settle down. I haven't noticed any audio issues from my end, and a couple other people indicated that audio is fine for them. So hopefully this is a temporary issue and we can continue. One thing
Omar,
to note that all the webinars are recorded and saved, and so if for some reason you cannot hear something, you will get a link to the YouTube recording of this webinar, which you will be able to view later. Sorry, David, go ahead.
No, I was just going to point out the same thing, that this was available as a YouTube video. So for anyone who has audio issues, they should be able to get back to that on YouTube. Sorry for the duplication.
No problem. Okay, so I just wanted to show this one more time. So this is out of the people who voted so far. This is the breakdown of the resources that people are using. Obviously, people can use more than one resource, and this is a multi-select poll. If you haven't responded to this poll yet, feel free to do so. You should have the link in your chat box, and you can go ahead and do that. And I will show this again towards the end of the webinar. So as David indicated, all of the databases that we put together are built on the same infrastructure. So they all look the same. So I'm just going to jump, for example, this is VectorBase. Here's TriTripDB, here's PlasmoDB, here's the VEuPathDB portal site, and they all look the same. What will be different between them is obviously the data content, and that's obviously an important distinction depending on what kind of tools and functionalities or tools and technologies are available in a particular organism. The home pages. So this is if you have been a user of the legacy VEuPathDB resources. This homepage looks different, obviously, and also if you've been a user of the legacy vector-based resource, this homepage looks very different. And so there've been a lot of changes, but the cool thing about this is that we've been able to merge some nice technologies from both resources into one, and they're all built on the same infrastructure, as I mentioned. We will highlight some of these features and some of the cool tools that are now available for everybody, and so those will be highlighted throughout this webinar. We have conducted multiple webinars over the last few months, and we will continue to do so. If you're interested in learning more about those, you can go under the help menu on the top of the page, go to learn about VEuPathDB, and you'll notice there's a webinars option here. When you click on explore, this will take you to our webinar page. And so this lists all the upcoming webinars. Today's webinar is here, and you can click on this and then go down and register it. But notice here, there's a whole list of previous webinars. So any of these webinars, if you click on any of these, it will take you down to the section for this webinar with links to handouts that were used during the webinar and a link to the YouTube recording of that webinar. So this is a nice way to historically go back and learn more about the resource. And for every webinar we run, we will be adding those to that webinar page. So the breakdown of the homepage is really, there are 3 or 4 main sections, basically. The top section is the banner section or the header section. This includes the logo of the database that you are on. There's a site search. This is a new functionality which will be demoed next. There are a number of menus at the bottom of the site search, including your strategies, and we will show you what these are. Access to all the specialized searches that are available within the resources, access to tools, your workspace. Obviously, we have a way for you to analyze data using Galaxy, your own primary RNA sequence or population data, which then you can port back into your favorite VEuPathDB resource for further analysis. And we have a whole webinar that was dedicated to this a few weeks ago, and you are welcome to view that recording. Access to the data, both the data sets that are within a particular resource, or access to downloading the genomes, for example. One important thing that I want to note here is that our resources, as many of you I think know already, are not geared towards just providing a place where you can download data, but rather our goal is to give you the tools to ask questions about the data and really change parameters to what you think is important for your research, and also to allow you to integrate the data together. So asking a question like, find me all genes that are annotated as a kinase, right? That's a question, but you may want to extend this and say, well, how many of these kinases are expressed during the trophozoite stage of Plasmodium in this case? Or how many of them are kinases that are in anopheles that are expressed during an infection with Plasmodium, right? And so those are types of questions that you can integrate and then develop some hypotheses that you can go back to the lab and test. If you're interested in exploring all the VEuPathDB resources, under About, there's a list of all of them, which you can link to all of them and explore them. There's a lot of help and various options for you to learn more about the resources. And importantly, as David mentioned, the Contact Us link is heavily used. We love to hear from you. So if you ever have questions about the resource, or in general, you're trying to understand something and you need our help, if we know the answer, we'll answer it. If we don't know the answer, we will try and help you or guide you to the right place to get an answer. I want to point out to the right side here, there's links to various social media outlets and also our YouTube channel. So this will help you with more learning, but also you can create an account and log in to these resources. You do not have to create an account to use the resources, which is great. So these are freely available. But creating an account allows you to do additional things like analyzing your own data, because then that requires you to save data under your profile and so forth. So there are other functionalities that you get by logging in, including you then can choose whether you want to sign up to any of our newsletters to hear what's new, what's coming new during in the different releases. In the central portion of the page, we have a number of specialized searches here, and we will go through these later on, at least during the different demos that are presented by my colleagues. The central portion here is an overview of the tools and functionalities in the site. So these are places where you can learn more about how to use the resources. And so there are little short tutorials with links to additional resources if you need more help. But the goal here is to give you a really quick vignette of what is phenotypic data in the resource in the various databases and what you can do with them. And so this is a really good starting point for you. So if you're new to the resources, I would recommend that you click on each of these links to get sort of a view or feel of what the resources provide. And if you scroll down to the bottom here, you'll notice there are more in-depth tutorials and exercises. These are multi-page PDFs that will walk you through in-depth particular tools and resources, and we will continue expanding these and adding more here. These are exercises that we use in our workshops, and so they're really geared towards in-depth analysis and training. And the last thing I'm going to mention here is that on the right there is a news and tweet section. So this allows you to access the most recent news. So if you're interested in knowing what's new in the most recent release of PlasmoDB, you can click here and get more information. And of course, we have sort of a live feed of our tweets, which gives you instant access to things that we are trying to inform you about. So I will stop here with just the general homepage, and I will switch now to Suzanne Warrenfeld, who will tell us about site search. So Suzanne, I'm making you the presenter. And you should be in control right now.
Okay, sounds great, thanks. So one very new useful tool is our site-wide search. It's located up here in the header and remains visible from any page, so you can scroll down or you can go to My Strategies and you'll still be able to initiate a site search from here. And how you use it is you enter an ID or a text term and the site search returns a categorized list of hits for your ID or term. There are built-in filters to the results, so you can analyze your results, and you can port the results to the strategy system to download or mine with any of our searches that access the results of our bioinformatic analysis. I know the question you are all asking is, how do I get to a gene page? So let's do that right here. You just enter an ID, and you can click enter, or you can use this little magnifying glass. And the box in the center of the results then features a link to a gene page when there's an exact match. So here we are then at the gene page for the nucleolar GTP binding protein 1. I can use this back button up here to go back to the results of my site search, the last site search that I did. Let's do a search now for a text term, and I'm going to use the word kinase. And what I'm taken to is a result page with all results matching kinase. There's a categorized list of results on the left, and then on the right are details. And I can open up this to see what fields matched my term. So the word kinase was found in EC descriptions, GoTerms, product description for the gene, and so on. And I can scroll through these results, but that's kind of cumbersome because I have 37,000 results. So we have built in some filtering features into this result summary. The first thing to notice is that the results are categorized, right? So for the term kinase was found in 36,000 gene records. It was also found in about 350 POP-set isolate records. And there was even one data set that had the term kinase associated with it. So let's say we're interested in only genes. I'm going to go ahead and filter based on that. And now I have 36,000 results instead of 37,000. There's two things to notice that happen now, right? I'm also now, because I filtered by genes, I have this gene-specific fields that I can filter on. And this export as a search strategy became active. It's only active when there's one record type showing in the result. Let's concentrate over here on the filters. And And let's say that I'm only interested in genes where the term kinase was found in the product description. I could go ahead and filter on that, click apply, and now I have about 16,000 genes instead of 37,000. And there's also an organism filter that I can use. So let's say that I'm interested only in T. brucei 927 genes, and I can filter on that and apply. And now I have a set of 271 genes instead of 16,000. I can clear all of these filters one at a time, or I can clear all of them at once from this clear filters up here in the input box. Let's go ahead and demo that. So I can go ahead and clear the TB927. I have 16,000 genes again. And let's say I was really interested in Lister genes. I'm go ahead and search for that and apply. And now I have 259 Lister genes. So the last thing to demo is this export to a search strategy. I can take all of these 259 genes and export them to our search strategy system. So they show up as a single step strategy with 259 genes that I found in the site search. These are Lister 427 genes with the term kinase in the product description. I can use this download button over here to create a file of these 259 genes. So here we are in the download tool. I can choose the top one, and I can download this list of genes with any data associated that I care to. Right? Click genes, and here's that download. I'm going back to my page, and I'm going to go back to my strategies with this link right here. And I'm back at my page with my single step strategy. Now the advantage here is that I can that I'm in the strategy system, and I can mine these genes with our searches that access the results of our bioinformatic results. So if I click add step, then I can access any of the results of our bioinformatic queries that we've run and add that to our kinase genes. I'll just demo that here, but somebody later on is going to go into this in more detail. So I can combine my kinase genes with other genes. I will intersect them with a search for signal peptide genes. And what I find is that 10 of my kinase genes are predicted to have signal peptides. Now, there's one important distinction here is that the search strategy system accesses the results of our bioinformatic analysis. So we have run the Signal P program and predicted through all of our organisms which ones have signal peptides. Up here, this is a text search that just looks for the term within the records. And you'll get very different results if you do that. So that's the site search. I think that's
great.
A great
Thank
overview.
you, Suzanne. I think there was a comment from one of the attendees that if we can make our screen sizes bigger, so we'll try and do that for the next section, which will be run by Evelina Bashenko. And Eve, I'm gonna, let's see if you're already the presenter. If not, I will make you the presenter. Oops. All right, Eve, you should be getting a presenter option right now, hopefully. And we can't hear you if you're speaking.
Yes. Can you hear me now?
Yes.
Okay,
good.
Excellent.
And yeah, so if you can increase, yeah, you did increase, I think. Go ahead. Okay.
Yeah, you should be able to see my screen now. All right, folks. We are on the FungyDB website, and I will run you through a quick search using the phenotypic evidence integrated in FungiDB. So first of all, to access the phenotypic evidence, you can essentially deploy two different searches. One using our menu from the top under the searches tab, where you could just start searching for the first words for phenotypic data and deploy the phenotype evidence search. Or you can also navigate to the left panel that says search for, and also start typing the first few characters, and I will deploy the search from here just based on my preference, but in any case it's accessible in various ways. So when you click on phenotypes evidence search, you're essentially redirected to a web page that contains a list of current datasets in FungiDB. And note that we have a combination of phenotypic datasets, such as those that are manually created by VEuPathDB or integrated from the labs, individual providers, or external databases such as Firebase. In this particular example, I will construct a search using Firebase annotations, and then I will show you how to run GoTerm enrichment analysis on your results. So let's go ahead and do that. To set up a search, I will click on the CP button right here on the right, which stands for Curated Phenotypes. And when you click on the button, the page on the bottom essentially brings up all available metadata for this particular dataset. Now note that Firebase dataset includes many organisms. So essentially you have a combination of disease-related phenotypes, whether it's pertaining host or different characteristics attributable to the pathogen itself. And in this particular case, I would like to look for all genes that have annotation of invasive aspergillosis. So I'll go ahead and use the filter search here and enter invasive first few letters, and here we are. We have essentially 205 genes that have invasive pulmonary aspergillosis annotated. So I will go ahead and select that data set and click Get Answer. So essentially what we have just performed, we deployed a search looking for any gene with that particular phenotype, and we have returned 131 genes. Now note that we have deployed this search without specifying any particular species. We just looked for the phenotype that is associated with invasive aspergillosis. So that technically would bring up potentially several Aspergillus species, including Fumigatus, Nidulans, or some others. Let's take a look at the gene list that we have. Notice that there is a results table right here, and that's essentially the list of genes that is being returned. And you also have an organism filter section on the left. So with the note that there's a total of 131 genes. Well, if I wanted to look a little bit further as to which organisms are returned in my search and how many genes per each organism, I would like to go ahead and actually sort of look at this table here. I could open up all the drop-down menus, but we have hundreds of genomes, so it will be quite tedious. So I will go ahead and say hide all 0 counts. So that's essentially eliminates any results that have 0 hits, and then I'll expand the remaining tree for Aspergillus species. Note that we have one gene that matches Aspergillus clavatus, one Nidulans, and we also have a few for Aspergillus fumigatus. The reference strain AF293 and also A1163. So let's go ahead and run a GoTerm enrichment analysis on these results. And I will go ahead and select the dataset for Aspergillus fumigatus because that seems to have quite a reasonable number of genes so that we can take a look and see if we can find something interesting. And obviously, you probably wouldn't want to run the Go enrichment analysis on one gene because that's essentially one gene, one Go term. But before we move on to actually getting the results for GoTerm enrichment analysis, let me show you how to deploy this search. GoTerm enrichment analysis is available from the Analyze Results section. If you click on the blue tab at the top, you essentially will be, the page will be refreshed and you will be presented with 3 different choices how you can analyze your results. We'll choose gene ontology enrichment, and the page will redirect you to a list of parameters that you normally choose to set up your search. And while the page is loading, I want to say a few words about gene ontology. What is gene ontology and why is it important to run the enrichment analysis? Well, first of all, gene ontology is a collection of terms or a system that have been implemented by Go Consortium, and it's an ability to describe our knowledge of a certain biological domain in 3 various aspects, such as biological process, molecular function, or cellular component. And what that means, you essentially could take a gene product and describe its molecular activity based on a particular function, for example, protein kinase. You can also take a gene product and characterize it using a cellular component, such as the location of this activity in relation to cellular structures, for example, cytoplasm. And finally, you can also take the gene product function and describe it in terms of biological process, such as where does this term where is this term occurring in relation to specific function, such as DNA repair or oxidation, etc. And today we'll run a gene enrichment analysis on biological process, and I will choose to run this on Aspergillus fumigatus AF293. Note, because we have returned a list of genes for various organisms, you can deploy analysis for each individual organism, but Go enrichment analysis can be only run at one organism at a time for statistical reasons. And before we'll deploy this search, there's a few things to take a look. First of all, at the type of evidence that we want to examine, I would like to run a full search using computed and curated data. Computed data is also referred as an annotation that is inferred from online resources or such as automated resources. Go Ontology has a dedicated website where you can read a little bit more about how various codes work. For example, the automated codes are often referred as IEA, such as inferred from electronic annotation, and most of the time the method that is being used is interpro to Go. But within the individual gene record pages, we do identify where the codes are coming from, and if you see any of the terms such as described here, this is a good website to refer as to how these codes have been produced, to explain a little bit more where the functional annotations is actually coming from. But going back to our strategy, so we will run a search using both computed and curated, such as manually created data. And in this particular case, I will not limit my search on GoSlim. So the difference between Go and GoSlim essentially gives you a snapshot of the major groups that occur in your enrichment. Imagine if you have a list of thousands of genes in your data set, and you don't necessarily want to look at you don't necessarily want to return every single GoID per subgroup. You may want to look at the sort of broader picture as to how this multi-thousand gene list fits within the let's say when you're comparing to another gene list obtained from a different sample or growth conditions. And what I mean by that, if let's take a look actually at the Go Ontology terms. So here's a list of Go Ontology terms that correspond to responses to stress. And if you scroll down, I am on the Gene Ontology website. If you scroll down, response to stress is a higher hierarchy term that includes essentially all of these Go terms. When you work with quite large data sets, it's quite useful to limit to Go Slim, meaning that all of these Go terms will be essentially condensed into one term, response to Go stress. And that can be quite useful, as I said, when you run quite large data sets in your analysis. So let's go ahead and proceed with our analysis using GoTerm enrichment, and we'll click on the submit button to deploy the analysis. Now, while the analysis is running, I would like to mention that within this interface, you can run multiple GoTerm enrichment analysis, and they will open up as separate tabs. So let's say you wanted to compare GoTerm analysis and Go Slim. So you have an option to run this side by side and save it under different names. So I pre-ran both analyses before, and here's an example of the GoTerm enrichment for biological processes. Now, one easy way to distinguish different types of gene ontology enrichment analysis that you run is actually give them a meaning, a name. You can rename a data set by essentially clicking on rename this analysis link right here at the top, and by giving your data set a descriptive title, you can differentiate between not only different types of Go enrichment analysis, but also if you choose to deploy GoTerm analysis, let's say on a different strain in Aspergillus fumigatus. Okay, so let's examine the results of the GoTerm enrichment analysis that I have run on the biological function. If you scroll down, every GoTerm enrichment analysis will essentially generate a table with Go IDs, Go terms, genes in the background, meaning genes in the background within the resource that has this particular Go ID associated, and also genes in your data set, such as amongst those 133 genes that have that description. This is also a percentage expression of this data, and you have a few parameters right here, such as fold enrichment, odds ratios, p-value, and a few other statistical correction values that may be useful. If you haven't listened to our previous webinars, check it out. But we went a little bit over in greater detail over these parameters. But in brief, fold enrichment is essentially the ratio of the proportion of genes in the list of interest with a specific Go term over the proportion of genes in the background with that term. Odds ratio determines if the odds of the Go term appearing in the list of interest are the same as that for the background list. And the p-values and especially the correction values are here to guide you to interpret the validity of the result. The p-values are generated as a result of the Fisher's tests, but for the very large scale data sets where essentially every single Go term is treated as individual entry and compared within the scale of genome, this ratio can be skewed, and as such we produce two correction factors known as Benjamini-Hochberg false discovery rate and also Bonferroni adjusted p-values, and that's essentially a method for correcting significance based on multiple comparisons. All of these tables are essentially scrollable, where you can sort based on the p-value and look at the highest hits, and interpret your results. The correction values quite come handy when you have to make a difference, whether a p-value may be sort of a borderline or outlier, to where you may need to clarify whether it's useful. But for those p-values that are sort of high quality and give you very good margin of confidence, essentially all p-values are very close to each other. So in this particular example, we have a list of we have a list of Go terms that can be visualized also by opening them in ReviGo. So ReviGo is a web server that essentially summarizes your list of terms and uses clustering algorithm that relies on semantic similarity measures. What it simply means, it looks at the Go terms, associated p-values, and just compiles them all together by taking all the redundant all redundant Go terms or those that can be grouped together, and provided you with a very nice output. I recommend reading some of the publications published by those who created ReviGo, but here is a very nice scatter plot that describes not only sort of the significant p-values, such as those in green and blue will be more significant than orange and yellow. It also shows you the proximity of various clustering, and one neat thing that it does for you is essentially takes all of the Go terms that belong to a larger group and combines them together in a simple group for easy to interpret.
Eve, sorry to interrupt. I want to make sure that we have a little bit of time left, and I'll make sure we get into some examples in vector-based. I think it's important to refer people to the previous webinar that we ran a couple of weeks ago on Go enrichment, and so there's a lot of details there, exactly what Eve is showing you right now. So if you're interested in learning more about Go enrichment, I would encourage you, as Eve suggested, to go to the webinar page, which we will link again in the chat window, and also you will get a follow-up email from this webinar, and we'll include the relevant links for you. Eve, was there anything else you wanted to say to wrap up before we switch over to Gloria?
No, that is it. I'm actually, that was my last sentence.
Okay, perfect. So I think we'll switch over to Gloria Geraldo Calderon, who will describe a search in vector-based. I think hopefully what you're appreciating right now is that the various databases allow you to run similar types of searches and similar functionality. And I think the cool thing here is that you can integrate between data sets, analyze data sets, visualize data sets. And so it's a one-stop shop where you can do a lot of different things. And I would encourage you that if you're ever trying to run an analysis, or you're finding yourself doing something repeatedly manually, check with us. We might have a tool already for you that will save you a lot of time. All right, Gloria, it's all yours.
Hi, Omar, can you hear me?
Yes, loud and clear.
And you can see my screen?
Yes.
Okay. Hello, everybody. So I'm going to give you just, a quick demo of how to construct a strategy. So Suzanne already used a keyword to start a search. She used kinase. Eve used phenotypic data, but I'm going to use a different example. What if you already have the list of genes that you want to investigate some information about? So you start typing here ID, and here I have them, gene IDs. Oops, sorry.
So as the page is loading, I wanted to say and remind everybody that these are beta sites, and beta sites typically are, a stage in the development of a new software, a new resource. And so as you use these sites, you might come across bugs, which is to be expected, but also it'll be great if you always click on the Contact Us link and say, oh, I tried to run the search and it gave me an error, right? And so that's always important for us to hear about. Sorry, go ahead, Gloria.
No, that's fine. Thank you, Omar. So here you have two options. You can either type the gene IDs, or you can upload a file. So I'm going to upload a file, which I have here. And again, it's just a text file, txt, with my gene IDs of interest. And I click on Get Answer. Okay, so it's exactly showing me what I was expecting. I uploaded a list with 11 genes. So for those genes, I can continue with my strategy. So I can change the name here, and I can say these are Anopheles gambiae genes, and these are opsins. And I can add a step to my strategy. So for example, I want to transform these genes, and I want to know the orthologs in another mosquito. For example, Aedes aegypti. So I click orthologs, and notice you have these drop-down menus here, but you can just type Aedes aegypti, you select it, and run step. And here you have it. So I started with 11 genes from Anopheles gambiae, and I got 12 orthologs in Aedes aegypti. If you want to check if you're actually done what you intended to do, notice how when I hover, I get these edit options. So I want to check, did I did what I was supposed to do? Oh yes, I was uploading the gene IDs, and here I selected Aedes aegypti, that is correct. But now you want to start looking at your data. So you come here, and this is, notice, these are the gene IDs from Aedes aegypti, AAEL. And these are the gene pages for each one of these records, where you will find all the information about them. So here is very handy for you if you use this navigation panel on the side. For example, you are interested in transcript data. So you're wondering if for this particular gene, is there any expression? And here's where you come to look for that, and you also have the data here. Okay, so, but that's when, that's when you start with a list of gene IDs. What if you don't have a list of gene IDs? So for example, you're interested in genes that have transmembrane domains. For example, you work with GPCRs, and you want to investigate them in all arthropods. So first example, I type a species. Now I want to select all the arthropods. And GPCRs have 7 transmembrane domains. So I change this, get my answer. And notice here is the search strategy that I just ran. And here at the bottom, I have the new one. Okay, so I have my 7 transmembrane domains. Now what I want to know, okay, because I'm interested in GPCRs, so I click here in transform, in combine, and I want to look for the interpro domain. And in the PFAM database, so if you're not familiar with this, so there is a database that combines all the databases about protein domains that is called Interpro, and one of those is PFAM. So I can select opsins, rhodopsin-like, that's the same opsins, run step. So, arthropods, run step. So what I did here, initial query, I wanted 7 transmembrane domains. And from those, I only wanted the ones that are, the category is rhodopsin-like. And what else can I do? I can add another step. So notice, I started with 6,494 genes. And the genes that have this interpro domain are 3,000. But in between these two, what they have in common is 1,529 genes. And if I'm going to quickly here, please remember that we had a special webinar about constructing search queries on May 7th. So you can go to our page of webinars and watch that recording. So what else I can do here? I can say, wait, I only want to see this. I don't want this to include chordates. I don't want this to include humans. So I only want this in arthropods. And I go to Metazoa. And notice if I click once, I get a check mark. But if I click twice, I get an X. And I see I'm going to the top of the hour, so I'm going to quickly wrap up, Omar.
Sounds good, not a problem. And as this is running, there was a question about how we determine percent similarity when we look at orthologs. So both this search that Gloria just showed us with the Orthology phylogenetic profile search and the Orthology transform, which she showed you in the previous search, rely on another resource that we put together called OrthoMCL. And OrthoMCL, essentially, it's running a BLAST similarity. And there's a lot more detail. I provided the link in the chat to the About page on OrthoMCL, which provides you the details about the similarities that are used and how OrthoMCL runs.
And I'm showing you here OrthoMCL. So here's the link, and this is how you can navigate to all the other sites. And this is the logo for OrthoMCL. Okay, so and as you know, same as Susan showed you, here you can filter the data. So I only want to see the data for Gambia, for example. So notice I start with, I have here 530 genes. So if I only select Gambia and I apply my filter, notice here I have this little icon telling me I have filtered the data and only I'm looking at 19 genes. Besides that, you can copy your queries just to modify them. You can share with your colleagues. You can save their strategies to my strategies. And here are all the other ones that I have in my panel. That's all I wanted to say, and just an invitation for all the vector-based users that next week we are going to have a specialized webinar for vector-based, because, you know, as Omar said at the beginning, this page may look way more different for you guys, and we are going to give a very detailed walkthrough of what genomes we have, transcriptomes data we have, and how to use all the new tools that we have. Thank you very much.
All right, thank you, Gloria. So I'll go ahead and make myself the presenter again. And make sure we have the right screen. Okay, so quickly again, just to wrap up a little bit, let me switch back to the browsers. So this is showing you the OrthoMCL website. And if you go to the About OrthoMCL, you'll find a lot more information here about this. And of course, send us questions if you have more questions. And just as a reminder, if you go under Help on any of our resources, you can go to Learn how to use VEuPathDB and then Explore Webinars. And as Gloria indicated, next week we have a special webinar that will focus on vector-based. So we will be running different kinds of searches, analysis tools, and so forth. So obviously, highly encourage vector-based or vector biologists to attend this webinar, but also any other person who uses VEuPathDB resources. This webinar will be relevant to you too, because we'll be describing and showing tools that are available everywhere, right, in all the resources. So we may be running an analysis on Anopheles gambiae, but you can run a similar analysis on Giardia, for example, or Cryptosporidium or Microsporidium, so or various fungi. And so so that's the nice thing about having resources that use a unified way of analysis. Another webinar I wanted to highlight, this is the one that's happening tomorrow, and this is about MapView. So this is our population biology viewer and analysis tool, right now available in vector-based. So this is clearly very useful for vector biologists, and something that we'll be making available on our other resources. But that's tomorrow, so feel free, obviously, to register for that webinar, which will take place at 10 a.m. US Eastern Time tomorrow. I'm going to show quickly the direct poll in the end. So if you voted on this, that's great. Thank you very much. And you can see the relative or general breakdown of website usage on the different resources. If you haven't voted yet, you can look in the chat box and you'll find a link to this poll. So go ahead and if you want to, you can go ahead and do that. Another very important and one important thing, I just saw a question about where I can view this webinar again. So this webinar is being recorded. As soon as the recording is done processing, we will upload it to YouTube, and tomorrow morning you will get a link Everybody who's attended the webinar will get a link to the recording. We'll probably add a link to the additional webinars. And also, you will get a link to the end of webinar survey. And so I'm putting it up here as well, and I will post the link in the chat box. So you can click on it and answer it as well. But it'll be very important. Oh, Sam did that already. Thank you. So it'll be very important if you go ahead and answer this survey and let us know how we did, but importantly, let us know what else you want to learn about. So definitely do this and do it soon. We know from experience that if you don't answer the survey right away, the chances of you answering it tomorrow or the day after decreases dramatically. So I'm going to see if there are any other questions. Maybe anybody else on the call. Are there any other questions? So if you have questions, go ahead and use the question answer panel. And I'm just looking through them quickly to see if anything new came in.
It looks like we do not have any new questions.
Okay, perfect. So thanks again, everybody. Thanks for all the presenters. And again, please stay tuned for additional webinars. We will repeat webinars as well. So if you miss something, we will continuously run these. We've been running them on a weekly basis right now, and we have many more webinars scheduled. Thank you for attending. Stay safe, and we look forward to seeing you online again. Take care, everybody.